We've got a serious subject today, but I think it's going to be a lot of fun, guys. The incidence of fungal infections is rising in immunocompromised people, and the morbidity and mortality of these infections are high. Recent threats include multi-drug resistant Candida auris. However, antifungal resistance is rising in other species as well, such as Aspergillus fumigatus. But have advances in diagnostic testing kept up with the accelerating threats of fungal infections? Welcome to Editors in Conversation. This episode is brought to you by the Journal of Clinical Microbiology, available at jcm.asm.org and on Twitter at jclinmicro. I'm JCM Editor-in-Chief Alex McAdam. This podcast is supported by the American Society for Microbiology, which publishes JCM. I'm joined by three experts to discuss gaps in the laboratory diagnosis of fungal infections. Dr. Esther Babady is the Director of the Clinical Microbiology Service at Memorial Sloan Kettering Cancer Center. Thanks for joining us, Esther.
Thanks for having me.
Also joining us is Dr. Shawn Zhang. Dr. Zhang is an Associate Professor of Pathology at the Johns Hopkins University School of Medicine, where he directs the Clinical Mycology Laboratory. Shawn, thanks for being on the podcast.
Thank you for having me.
And we're also joined by Dr. Shawn Lockhart. Dr. Lockhart is the Senior Clinical Laboratory Advisor at the Centers for Disease Control. Shawn, I'm pleased you could join us.
Great to be here. Thank you.
Esther, the three of you created a group called the Fungal Diagnostics Laboratories Consortium. The consortium wrote a paper that's in the current issue of JCM, and we'll come to that in just a few minutes. First, can you tell us about how the consortium began?
Yes, happy to. And thanks for asking and again for having us here. So as you mentioned in the introduction, I'm the director of the microlab at Memorial Sloan Kettering Cancer Center. So my laboratory deals a lot with immunocompromised patients, particularly patients with hematologic malignancy and transplant patients. So while we have been really good at doing diagnostic for bacterial and viral infection, fungal infection has remained quite of a challenge. And when you think about this immunocompromised patient population, as you mentioned in your introduction, fungal infection is a very serious disease, and it's creating a lot of challenges for us with an increased mortality and morbidity. So this is something I've definitely been interested in solving as a problem of fungal infection. In 2018, ASM actually organized this conference called the Clean Micro Open. And the goal of Clean Micro Open was really to bring lab director and industry partner to identify gaps in the laboratory in clinical microbiology lab and how to solve them. So you had to apply for a position, or so to speak, in the Clean Micro Open by writing a proposal saying, this is a problem we've identified, this is how I think we can solve it, and this is how industry can help. So of course, I wrote a proposal for fungal diagnostics. And so the meeting was in West Palm Beach. And when I arrived there, Shawn Zhang from Hopkins had also submitted a proposal for fungal diagnostic as a gap in diagnostics. So we both presented we both did our presentation and had a little spiel about why this is important and why we need to fix this problem. And so we broke into this work group during the Clean Micro Open. And when we came back together, the work group had to decide what we were going to be doing to solve this issue. And both Shawn groups and my group brought up the issue or the idea of forming a consortium. And the reason for that is while the number of immunocompromised patients is increasing, the overall number, when you think about all the other patients, is still relatively small. So we had to treat fungal infection as either a rare disease or a neglected disease. And how you deal with that is by forming collaboration and creating networks so that you get more strength in, small number at Sloan, small number at Hopkins, but overall that becomes a big group of patients that we can investigate. And for the industry partner, that's sort of the idea is like, if I'm going to invest a lot of money in developing tools and tests, there should be a market for that. So the idea of the consortium came about. And this was in May. We fast forward a little bit, a few months later, Shawn is at ID Week, and this is Shawn Zhang. He's at ID Week and he's presenting a talk. And at the end of his talk, he had a slide about this idea of the consortium that came out during the Clean Micro Open. And so Shawn Lockhart now reached out to Shawn Zhang, say, hey, I have the same idea. Why don't we all work together? So by October, the two Shawn and I have to keep saying Shawn Lockhart. The two Shawn and I got on a conference call. So this is pre-pandemic, so Zoom wasn't really a thing. And so we had to get on a conference call and discuss the idea that we've had at Clean Micro Open, and that Shawn also had that idea separately, Shawn Lockhart, that is. So we got together, started discussing what exactly we wanted to tackle, how we were going to operationalize it, and who was going to be invited. And we wrote a letter with the ideas and the goals, and reached out to a lot of our colleagues, and primarily to those who dealt with a lot of immunocompromised patients. Again, the same idea of strength in number. And the response was pretty amazing. Everyone that we reached out to was really interested in joining the consortium. So we invited over 20 centers, and we had our first meeting at Microbe in 2019. And it was really exciting to see how this went from an idea at Clean Microbe the year before to a room full of experts, really, a diagnostic expert from like top-notch university or an academic center and hospital already interested in solving this gap in fungal diagnostics. So that's really how we all got together, and very excited about how things ended up and how we're moving forward with this.
I think it's great. You know, you hear about this kind of thing and people talk about doing this kind of thing pretty frequently, but this one has really come to fruition.
Absolutely, yeah.
Esther, I don't know if you remember, but at the end of that microbe, someone, I don't remember who, told me about your group that had formed.
Yeah.
And I thought I need to contact them because I want whatever they produce to be in JCM.
Yeah.
And I think I emailed you from the airport.
Yes, I remember exactly. Like, I want to say this in JCM as a mini review. So you give us some incentive to really get working as well. So
Good. Well, I'm glad it worked out.
Yeah.
Shawn, and they know which Shawn it is because they had these questions ahead of time. Shawn, can you tell us a little bit about the goals of the consortium?
Yeah, absolutely. Yeah, thank you for inviting us, and thank you for the questions. I think so. Basically, the consortium is trying to promote research collaboration across the centers that will support the development and evaluation of the new methods for improving the diagnosis of fungal diseases, and also identification and the detection of newer emerging fungal pathogens, and also collaboration on the antifungal drug resistant detection and the new drug development. So overall, the consortium is really providing a platform which will allow laboratories to join forces to facilitate and accelerate the research and the development to improve and enhance the fungal diagnostics. And then the consortium is also would like to also promote the collaboration with the industry partners to support the new assay development and the commercialization, as well as to accelerate the process for the clinical validation and the laboratory implementations.
Thank you, Shawn. I think it's interesting and valuable that you have linked up with manufacturers because in the US, that's how diagnostic tests become widely available. So I think that will be a valuable piece of this. Let's talk for a little bit about your commentary article in JCM. The title of the article is Recognition of Diagnostic Gaps for Laboratory Diagnosis of Fungal Diseases, Expert Opinion from the Fungal Diagnostics Laboratories Consortium. And we'll put a link to that article in the notes for the show. In the paper, you identified 6 disease-specific gaps and 6 methodological gaps in fungal diagnostics. So, Shawn Lockhart, to start, what was the process for selecting these disease-specific and methodological gaps?
Yeah, so that came out of our first meeting at Microbe when we all met together. And one of the goals was first to put together a survey just to see what was offered in the US. These were mostly biggest labs. The labs that I had pulled in were a lot of the reference and diagnostic, larger, even commercial diagnostic laboratories. And lucky for me, Shawn and Esther had reached out preliminarily to their clinical colleagues. And so we brought this large group together and we did a survey of what services were offered. And at the end of that survey, there was an open question: what are the 5 biggest diagnostic gaps that you see? And out of the 26 surveys that we got, it was really surprising how much overlap we had in those answers. We expected we could have had 50 different answers with 5 times 26 groups, and a little bit of overlap. But it turns out everyone was seeing the same big picture, and we all had seen these same gaps, which means they were glaring gaps, they were overwhelming gaps. And this survey that we did and the answers that we got really put into perspective how we saw it across the US and how we realized that we're all having the same problems, we're all seeing the same gaps, and we're all looking for the same answers. So it came out of that survey, and we were really surprised at how much on the same page both the big reference laboratories and the smaller hospital laboratories all saw things in the same light.
Thank you, Shawn. And I reread the article this morning, and I would recommend that everyone take a look at it, because each of the items that you identified is a serious gap. And each time I thought, yep, that's right. That's something we really need to have progress on. But we don't have time to go through all of it. I'd like to ask each of you to select one of the disease-specific gaps and one of the methodological gaps, and tell us a little bit about the significance of that gap and how it might be addressed. So we'll start with the disease-specific gaps, and we'll begin with you, Esther. Which one would you choose?
So for me, when I think about the main gap, pneumocystis pneumonia comes to mind, right? So PCP. And part of the reason for that is that I think about PCP as a relatively low-hanging fruit in terms of something that we can really solve by really getting together and focusing a little bit. So pneumocystis pneumonia is now an infection that traditionally used to be thought of a AIDS-defining infection, right? So you told us to tell story and tell jokes. Something that comes to mind is one way that I really like to relax is to just watch a lot of TV. So one of the things I've been watching recently is ER on Hulu. And ER is this medical show from, I want to say it was the late 90s, I think, 1990s. So when you look at the first few seasons of ER, it's still at that time where HIV was a big deal, right? So one of the very few episodes in ER, this patient comes into the hospital and then they collect his sample, they go in the back ER lab, which I thought was very cool,
Yeah.
of course, and they do a smear and they come out and they say, this patient has pneumocystis. And so before they even knew that he had HIV, they figure because he has pneumocystis, he has HIV. Mm-hmm. And so this is a show from over 20 years ago. Now, pneumocystis may still be an issue with HIV patients, but we see it more in non-HIV immunocompromised patients, like the cancer patient that I see at Sloan Kettering. When we think about the diagnostic, though, we still do that smear that they were doing in the back of the ER lab, and there is still a challenge in how do we really make sure that we can first diagnose that infection, and second, to make sure that because pneumocystis, like a lot of fungi, can colonize the host, how do we distinguish between infection and disease? And so we have done studies and we've looked at molecular testing for that. And right now, it's interesting enough that most labs are still using either calcium white or taurine blue, or if they fancy, they would use like a DFA, but not so much molecular diagnostics. And this is something that when you think about industry partner, can we develop a molecular test that can be standardized and used across laboratory to really solve this issue? I think it's a thing that's really simple to do when you think about it, but we just need to get organized and get that down. So I would say pneumocystis is, for me, a gap that we can tackle and hopefully solve relatively easy.
One of the concerns with pneumocystis PCR that I hear frequently, and that you mentioned in your article, is that people are colonized with pneumocystis, as well as are getting sick from pneumocystis infection. So if we did a nucleic acid amplified test like PCR, how could we distinguish it between colonization and infection?
Yeah, and I think that's been one of the reason it hasn't taken off so much, because that is definitely a question. One of the things we have even proposed in the article is thinking about not just developing method, but coming up with algorithm on how best to use them, right? So for all testing, you want to start with like the pre-test probability being really high. So we will provide information on which patient population should really be tested with that. But molecular testing can be developed and made in such a way that we would identify maybe thresholds across patient population that could distinguish between colonization and actual infection. But going back to the purpose of the consortium, this is not something that you can really do really well with a small patient population at a single center. Doing multi-center studies and looking at where we would set up this threshold and how we would interpret that, I think is going to be very, very important. And if we take an example from the viral diagnostic community, the same thing is true of like CMV, for example, right? We do quantitative CMV assay to monitor CMV viremia. And until we had diagnostic commercial assay available, this threshold from one center to another was very different, and they were not, you couldn't really compare them. So the idea would be like here, if we can start from the beginning, developing an assay that is going to be standardized and then coming up with threshold as we implement this, it would really be helpful in different center using an established and validated threshold to interpret pneumocystis PCR assays.
Thank you, Esther. And Shawn, Zhang, what would you identify as the disease-specific gap that you'd like to discuss that particularly interests you?
Sure. I would probably pick up the mucomycosis, so also formerly called zygomycosis. So this is a fungal disease. At this point, is not as frequent as the invasive aspergillosis. However, the clinical outcome for the mucomycosis is very often very, very poor. So in terms of the diagnosis for this disease, currently, the only approach we have available in the laboratory is the culture-based method, plus sometimes histopath examination. If there's tissue, for example, if there's tissue specimen sent to the pathologist for examination. And often by the time when we made the diagnosis based on the culture, is often it's too late to actually change the clinical outcomes. So it's really, this is something we really need to improve the diagnosis for this disease. There is some very encouraging data come out demonstrating the mucolatus DNA, they actually can be detected as early as like 9 days before the diagnosis made by cultures. So that's really encouraging data. At the amount of the DNA circulating in those patients with mucomycosis is actually very, very high, higher than the amount of for example, compared with aspergillosis DNA. So this is really I think it's exciting, and also will push the early diagnosis. So I think that comes to the molecular approach, which is the nucleic acid amplification test to detecting those mucorales DNA in those patients' serum or blood specimen. However, there's very scant data actually published on the molecular assays, and if you look at the most recent guidelines, which is published in Lancet Infectious Disease, the global guideline for mucomycosis, the diagnosis and management, the evidence supporting using the nucleic acid amplification test is not very strong. Is not very strong because of lack of sufficient data to support. And I think that one of the major hurdle here is lack of standardizations of the methods, molecular methods in the clinical laboratory to improve the speed and the sensitivity for diagnosis. So, for example, in terms of the sample types, extraction methods, the gene target, all those variables will actually contributing to the performance of the molecular assays. So on the other hand, a commercial test somehow can actually providing a standardized approach to overcome those variables. But to my knowledge, at this point, there's only one commercial assay available for detecting the mucorales DNA in the clinical specimen. And for that commercial test, it has not been FDA cleared. So it seems there's lots of work to do. So I think for the consortiums, we think this is actually a gap, and we want to really have a multi-center investigation seeking to standardize the mucorales nucleic acid amplification test and assess its clinical utilities and can actually improve the diagnosis of this rapidly progressive and often fatal infections.
Thank you, Shawn. And finally, Shawn Lockhart, which of the disease-specific gaps would you pick out as one that particularly needs to be addressed?
So I'm going to cheat a little, and I'm going to actually pick two. And the first one I'm going to talk about is one that's unfortunately near and dear to my heart, and that's Candidemia, because I started studying Candidemia in the early 90s as a postdoc because my mother-in-law died of a disseminated Candida infection. And so that really got me into the show, and I've been studying it ever since, despite the fact that we've only been able to detect about 50% of candidemias based on autopsy studies. Nothing has really improved in our detection systems. We're still using the same blood culture we've been using for years and years. We're still probably only detecting 50%. And because of some really nice studies that came out of our colleagues in Pittsburgh, we know that we're missing a number of deep-seated infections in Candidemia. Now, this is the number 3 cause of bloodstream infection in hospitals, and yet we're only detecting 50% of those infections. We've got to find a way to really improve on that and make a better system so that we can, number 1, detect more of the Candidemias, and number 2, detect those deep-seated infections that we're definitely missing. The other one that I think we're actually making improvements on, but there are some other problems, are the endemics. And, being in public health, we do a lot of studies on endemics, and they're, kind of ignored by most other places. But in public health, it's a big problem for us. And the good thing about endemics is that we are now increasing the number of tests available. There are becoming a number of commercially available tests, antigen and antibody detection tests for histoplasma, blasto, and coccidioides. The problem lies in the fact that we don't have specimens to validate. So if you cannot validate these tests in your laboratory, how can you bring them on board? And one of the things I think the consortium would love to do is set up some sort of biorepository. If we each get 2 histo and we all contribute them to this biorepository, suddenly we have 50 histopositive serums or cocci positive, and we set up this biorepository that becomes available as a tool for anybody who wants to bring on these new diagnostic tests in their laboratories, any commercial entities that wants to develop a test. So putting our resources together and working towards these, getting this common resource, I think would be fantastic for solving some of these gaps.
Thank you, Shawn. At least a couple of times we have heard about nucleic acid amplified tests and the need for nucleic acid amplified tests. I'm just going to throw this out to the three of you. Anybody jump in? It seems like the virologists have been playing in this yard for a long time, this playground. They were the first in, I think, on nucleic acid amplified tests. Bacteriology is all in on nucleic acid amplified tests. What's up with mycology? Why is it a little bit behind?
So I'll actually tell you a funny story about that. So you remember fungal meningitis a few years ago, exorahilum, in all those patients. We had tried to get it out of the CSF by using nucleic acid amplification. We used the old fungal tests. We spun out the CSF. We took the pellet and we tried to amplify it, and nothing worked. We weren't detecting exorahilum. So what we did is we thought, well, we know there's white blood cells in there. They must be busting open the fungi. There's got to be free DNA. Let's get the virology kit. And we literally turned to the virology kit to collect the free DNA in the sera. And that's what we used to develop our tests to detect exorahilum in all of these fungal meningitis patients. So yeah, jumping over to those virology folks, they seem to be going in the right direction, and perhaps we have something to learn from them.
It all points back to virology. Esther and Shawn, Zhang, anything to add?
No, I think that, it's interesting that you went to the virologist to do that. Just because when I think of some of the challenge with fungal is, viruses are small, they're easy, you manipulate them. You have to worry about RNA versus DNA. But with fungi, the fact that they're eukaryotic organisms and you have to actually process them a little bit differently than you would a sample for virology or bacteria. I mean, bacteria, you don't even have to extract them sometimes to actually get enough nucleic acid. I think the challenges are a little bit different, and you had to really get invested in it to get this assay developed and optimized. And then the other thing is, one of the things that has been challenging with fungal diagnostic using molecular is the fact that, this is a fungi world and we just live in it, I think, like Aspergillus in the lab. I mean, Aspergillus in your house, like you see it, right? And so being very careful, using a method that's meant to amplify things extensively and then worrying about just environmental contamination, it brings a different level of challenge into the use of nucleic acid for that purpose. Going back again to wanting to standardize the approach and standardize how we do this, to make sure that we don't start diagnosing, colonization versus infection.
Thank you, Esther. And we've started to move towards it already. So if it's all right, let's continue to go to the methodological gaps. And again, each of you, if you could pull out one of the things from the article, and we'll start with you, Esther. Which of the methodological gaps jumped out to you?
Yeah, and so one of the things I do love to do is molecular diagnostics. So, I'm going to pick something related to molecular. And for this, the identification of mold from FFPE, so from formalin fixed paraffin-embedded tissue, I think is one that we identify as a gap in the article. And I think we've all had that experience, right, where a patient goes to the OR or go to interventional radiology, biopsy is done, tissue is collected and sent straight to pathology, where they drop it in the formalin bowl, and that's the end of it, until they do special stain and see that there are a fungal element, and then finally call micro to find out if there was tissue for culture. And of course, there was no tissue. And so because that's a scenario that happens often enough, and for me, again, at a cancer hospital where they do biopsy, they're not thinking, COX-D or histo, they're thinking, some sort of cancer. And then this is an incidental finding. How can we use that tissue to come up with a diagnostic, since just knowing that it's fungal element is not enough, and making the differentiation between Fusarium and Aspergillus in a tissue might not be so straightforward. So again, going back to that, how do we standardize this? How do we determine which gene target is the optimal one? How do we determine, do you do it only when you see fungal element on the tissue in special stain, or can you attempt this even when there's nothing there, but there's a high suspicion of the infection? And so fungal diagnostic FFP, I think there's only a handful of labs in the US that have that methodology set up and done routinely. And, most of us are on ClinMicroNet. We know, right? Like, where do we send? Like, I have this, where do I send it to? And those are the two labs. There's a couple of labs. So we're thinking that definitely coming up with a standardized approach and protocol to doing that, from FFP to doing fungal diagnostic from FFP tissue is a gap. And as we move, this is now 2021, next generation. I mean, you can't talk about sequencing without mentioning next gen sequencing. That idea that maybe using next gen sequencing either on FFP or cell-free DNA in sterile sample would also be another approach for us to tackle this issue of fungal diagnostics.
Thank you, Esther. And Shawn Zhang, we'll turn to you next. Which of the methodological gaps would you like to tell us about?
Sure. I would like to pick up MALDI-TOF mass spectrometries.
Good. I was hoping one of you would pick that. Yeah, that's great.
And particularly for the modes, as you see the MALDI-TOF mass spectrometry. I mean, on the bacterial side, so let's pretty much phase out all the biochemicals and using the MALDI-TOF. And this is also same for the yeast. For majority of the yeast, now you can reliably identify by MALDI. However, if you look at the application of the MALDI on the modes, on the filamentous fungi, there's very, very few lab is actually implementing this approach. So I think overall, I think there's two big gaps, barriers preventing the clinical laboratory using the MALDI for mode identification. One is the extractions, because the modes extraction is not as simple as for bacterial and yeast. Because if you remember, if they're filamentous modes, they actually they grow as with hyphae, and they grow with spores, and different age of the colonies, and grow on different medium. And if you extracting those ones, they will probably generating different spectrums. And also the methods, if you're using the methods for extraction, can be also different. Some methods recommended you can use directly on the colony from the culture plates, whereas the Bruker systems is recommending using the liquid culture. Basically, they try to converting everything to the hyphae mass for the extractions. So and as I mentioned earlier, the different extraction methods, they were generating different spectrums. And if you're using one particular extraction method, and you have to and the database, if the database is built up on the one particular extraction method, and you have to using the same methods, otherwise your spectrum generating by different method may not match well with your database. And this has been demonstrating by the publication previously by the NIH group, which they develop their own database using the extraction directly from the colony grew on the culture plates. If they're using that spectrum compared to the Bruker database, which is generating by the liquid culture medium, they don't actually match very well. So that's actually very good examples. The second barrier hurdle here is the database, because if you look at the two commercial systems, one is the Bruker MALDI-TOF system versus the Vitek mass spectrometry systems. the database for the modes, filamentous mode, is very limited. So very often, if you do the MALDI identification of modes, 50% of the time you don't get the results because you don't have the entries in the database. So I think so as the consortium, we want to join forces, try to address those gaps. I think particularly for the database, we really want to push a kind of the free distribution of a continuously updated and a curated database, which like each clinical laboratory can actually use. So that will actually provide a significant benefit for the clinical laboratory to implementing this technology to identify the filamentous fungi, the modes.
Thank you, Shawn. And I have to say that the first point you made about the variability of the profile on MALDI-TOF, depending on the media, the age of the growth, and so on, is something that I hadn't really appreciated as well as I should have until I read your article. It's really quite striking. We see a little bit of that with bacteria, but far less than with fungi. And finally, Shawn, what methodological gap do you see as particularly urgent, and why?
I see antifungal susceptibility testing for molds as a really big urgent gap. So even when we did our survey amongst some of the biggest fungal laboratories in the U.S., only a handful of those laboratories actually perform susceptibility testing for molds. Most of them sent out to a commercial laboratory, and so that's a huge gap. There are no commercially available systems for mold antifungal susceptibility testing. There is a standard M38 by CLSI, but there aren't any commercially available platforms, and that's a gap. Now, it didn't necessarily form a huge gap in the past because of the fact that we only had two classes of antifungals essentially that were mold active, azoles and amphotericin B. So if one doesn't work, you only have one other choice. Why perform susceptibility testing? It's not like you can choose from 3 or 4 other antifungals to use on your patient. But now we're in an era that's really unprecedented. We have 2 new classes, not just 2 new antifungals, but 2 new classes of antifungals that are mold active, that are in phase 2 and or phase 3 trials right now, and a couple more that are just slightly behind that in development. So we could reach an era where we could possibly have 4 or 5 different classes of mold active antifungals available, but no way to test them. And so really, if we want to optimize how we're treating our patients, we've got to be able to perform antifungal susceptibility testing and really optimize treatment for our patient population once these new antifungals come online. And it's only a matter of time. I think we need to get a system in place ahead of time instead of, dealing with this wonderful problem of having multiple antifungals after the fact.
Thank you, Shawn. Esther, what do you see as the next steps for the Fungal Diagnostic Laboratory Consortium? You touch on this in your article. What do you see as the highest priorities?
Yes. So in the article, we mentioned what the gaps were. And there are 26 labs in the consortium. So now the question was, do we tackle all of them all at once? And of course not. So we again did what we did after we had our initial meeting and sent a survey for the group to rank those gaps. So this is what we've identified. So everyone just ranked from 1 to 5 what we should tackle first. So we sent a survey a couple months ago, guys. I can't remember, like a few weeks ago, and finally have all those results. And we've identified the top 5 or 6 priority, and many of those we've actually spoken about in this podcast. So the goal is that we're going to go back to the consortium. We've identified the top 5, form small group of people that are interested in that particular gap or that particular disease or method gap, and start working on it. If it's a method, reach out to an industry partner and try to figure out, how can we work together to develop a test? If it's a antifungal, how do we train people to do that? Because one of the things we didn't really discuss, education was also part of what the consortium wants to be, training of the next generation of fungal expert. So the goal really is, now that we've identified this, to form the groups and start tackling these individual problems and coming up with solutions.
Thank you, Esther. Either of the Shawns? Yeah. Shawn Lockhart.
So one of the things that came out of you allowing us to put that article is that, obviously it's being seen, and it was recently seen by a Canadian group that funds basic research in biology, and they have already reached out to us, their group to our group, and saying, let's sit down and talk about how basic research can work towards helping you fill those diagnostic gaps in clinical. So bringing the two worlds together, how can we bridge and utilize basic research to help us solve these clinical diagnostic problems that we're having? And so that's the other thing that we'd like to do is encourage people outside of our group, encourage basic research, encourage industry to approach us if they've got a problem or a solution that they, like our input. We don't want this to be this, tight group that just talks to ourselves. We want to advocate for fungal diagnostics wherever we can. And by you allowing us to publish that article, it was a great opportunity for us, and hopefully more groups will reach out.
Thank you, Shawn. So collaborations and possibly support coming from publishing in JCM happens all the time, folks. Shawn, Zhang, anything to add?
No, I think I would just want to say thank you for JCM to allow publish our position paper and also have this podcast. Really, actually, advocating our consortium group here. I think we like Shawn said, we don't want to just localize ourselves within the group. We really want to put this consortium as a platform which will allow people, the industry partners to collaborating with us, and also the basic research group, we really want to work them all together. Ultimately, our goal is to really improving the fungal diagnostics, saving our patients.
Thank you, Shawn. And I have to say, I think it's obvious, I'm really enthusiastic about this article and about the consortium. So I'm delighted that you submitted that paper to JCM. And now the part that Esther expressed a little bit of trepidation about. And
okay.
And that is the little quiz, the little game we do at the end here. And I'm basing this on an article I saw in NPR. It was on the radio first, obviously, but they published it on their website. I think Slate wrote about this. So it was published in a couple of places and picked up. In German, new words can be created by compounding 2 or 3 words together. And you won't be surprised to learn that several new words have come out of the coronavirus pandemic. The Leibniz Institute for the German Language has compiled more than 1,200 new words related to the coronavirus pandemic.
Oh my God.
I have used their list on the internet to find several of these. So I picked out 3 of these. I will read you the word in German, which I do not speak. I will read you the word in German, and then I'll give you 3 choices, and you can work together to try to figure out which of these is the correct definition of the new word. So here's the first one. Maskenmuffel. Maskenmuffel. Does that mean mask face, mask grouch, or mask voice?
Oh, I would say mask muffle. So I would go with mask voice.
I would want to agree with my voice.
Shawn's nodding. Yep. All right.
Same here.
It was mask grouch.
Oh.
What is mask grouch anyway?
Mask grouch. Somebody who's crabby because they have to wear a mask. I think we have all met people like that. Maybe we have them in our family. All right. Well, you have missed one, but you have 2 more chances. And if you get 2 of these correct, you get awesome bragging rights. Here is your next one. Klopapier hamstarn. Klopapier hamstarn. Toilet paper regulator, toilet paper thief, or toilet paper hamster?
It has to be teeth, right? Like, what's the point? We didn't have enough of them. People were stealing them at the beginning. I don't know.
I'll agree with Esther.
Are you sure you want to go with that one?
I
I'll read
think
it, I'll
I
read
would,
it to you. Yeah,
I would go with toilet paper regulator.
regulator. Huh?
Same idea.
Clopapier hamstarn.
I mean, I meant hamster. Did I say that? Did I pronounce that wrong?
You're correct. Toilet paper hamster. Hamster here refers to acting like an animal, acting without sense, acting without thought. So it's someone who hoards, toilet paper mindlessly. Excellent. You are on solid ground now. Here is the last one. Balkon Gessen. Balkon Gessen. Balcony singing, balcony dancing, or balcony drinking. Of course, we were all in our homes. People were doing things out on their balcony because they had been trapped inside all day. So is this balcony singing, balcony dancing, or balcony drinking?
Shawn, what do you think?
I was just going to say that.
Balcony drinking.
Yes.
Oh, I'm sorry. It's balcony singing. These were really tough. I mean, it was really just a matter of guessing. And I think you guys did a great job. So thanks to each of you. I have really enjoyed this very much. The new paper on gaps in fungal diagnostics is in the current issue of JCM, which is at jcm.asm.org. Thanks for listening.
Thanks.
Thank you.
Thank you.